learnt about primary and secondary active transport system, and we understood that both these systems, which are called as primary active and secondary active transport, they require energy for transporting their solutes from a region of low concentration to a region of high concentration, and that we call it as against the concentration gradient. And we understood that primary active transport requires hydrolysis of ATP, that is breakdown of ATP, and the energy which is released when ATP is broken down, that is used for transport of the solute in case of primary active transport. While in case of secondary active transport, we have a proton motive force, and this proton motive force is developed on the membrane surface because of a previous transport, and most likely the previous transport is a primary active transport. Fine. And we have examples of these two transport systems. One of the transport system is called as Na+K ATPase. This is actually a transporter protein which is transporting Na, that is your sodium ions and your potassium ions, and this particular transport system requires energy which is provided by the hydrolysis of ATP. So we call it as primary active transport. Then in case of secondary active transport, we have an example of lactose permease. This particular transporter protein which is present on your plasma membrane is going to transport lactose inside the cell, and for that it is dependent on the proton motive force which was generated on the membrane because of a previously established gradient. Okay, so these concepts of primary and secondary active transport are very important, and I hope it is clear to all of you. Moving ahead, in exam, if they ask for a diagrammatically representation, then you are going to go for this particular diagram which is there in Leninger. Okay, this is the diagram that you need to follow. As we can see, the first diagram is of primary active transport and the second one is of a secondary active transport. So as we can see, this is a cell and the cell membrane, and here you can see the transporter protein over here. The solute which is going to get transported is this orange color balls which we can see here. These are the solutes. As we can see, the concentration is high outside the cell and it is low inside. So during the primary active transport, the solute will be transported from inside to outside, and during this process we can see we have hydrolysis of ATP. So ATP की जो एक phosphate है यहाँ पे वो release हो जाएगा, and that energy is used for the transport of the solute from a low concentration region to a high concentration region. Okay, this is your primary active transport. Next comes our secondary active transport. So secondary active transport cannot occur on itself. There has to be some primary active transport because of which a membrane potential is generated. Okay, so we can see here this particular transport protein is the one which undergoes a primary active transport. Your solute, this S solute, will be transported outside the cell, and we have the hydrolysis of ATP over here. This is exactly same what we studied here. Okay, ये जो चीजें है, this particular transport over here, this is exactly same as the primary active transport here. Now what happens? This particular solute will try to enter your cell through this particular transporter which is undergoing a secondary active transport. Now along with this molecule, your solute also will enter the cell. Some other solute is called as S2. Okay, so two of them is going to enter the cell. As we can see, your solute is going from a region of low concentration to a region of high concentration. So this is called as your secondary active transport system. Today we will learn about 1.3.5 that is about the Na+K+ ATPase. Okay, so let's start with this. This is the diagram which is there in your Leninger. You have to refer Leninger for this. And this diagram is the one that you will draw in your exam. Okay, for the Na+K+ ATPase. So this square thing is nothing but your cell. Okay, let me discuss, make you understand this particular diagram. Look here, this square thing over here is your cell, and this dark blue shade region is the cell membrane. Okay, and in the cell membrane here you can see some molecule is over here. This molecule is the transporter protein, which is called as Na+K+ ATPase, and it is going to transport Na+ and K+ ions. Okay. First let us understand the concept and then we will come to the diagram. Fine. Now if you consider animal cells, so we are speaking about any animal cells, what a cell does is, cell always keeps the concentration of Na+ lower as compared to the surrounding medium. So cell wants that the concentration of Na+ is always low inside the cell and it should be high outside, but the concentration of K+ should be high inside the cell. Got it? That is the difference and the main important concept of Na+K+ ATPase. What the cell wants? Cell क्या चाहती है कि Na+ का जो concentration है cell के अंदर हमेशा low होना चाहिए as compared to outside, but the concentration of K+ should be high inside the cell. Got it? Na+ high कहाँ पे होगा? Outside, and low कहाँ होगा? Inside. While K+ high कहाँ होगा? Inside, and low किधर होगा? Outside. ठीक है? तो ये जो imbalance है, इस imbalance, why it is imbalance? Because one of the solute is high outside and low inside, while other one is low outside and high inside. तो ये जो imbalance है, इस imbalance को maintain करना पड़ता है. Because if there is something happens and this goes opposite, then there will be a problem. So we require some system to maintain this. Maintain क्या करना है? आपको Na+ को लो सेल के अंदर लो रखना है, लेकिन K+ को हाई रखना है. इसे मेंटेन करने के लिए आपके पास यहाँ पे प्राइमरी एक्टिव ट्रांसपोर्ट सिस्टम है using the enzyme which is called as Na+K+ ATPase. Now, why this is called as an enzyme? Basically, this is a protein transporter. ठीक है? It is a protein transporter, but it is having a domain or a region where it can undergo the ATPase reaction. ATPase is मतलब ATP hydrolysis. ATPase का क्या मतलब होता है? It means ATP breakdown or ATP hydrolysis. So ये जो एंजाइम है ये not just ट्रांसपोर्ट करता है Na+ और K+, but it is also capable of breaking down your ATP to form ADP and PI, and that is the action of an enzyme called as ATPase. So this particular protein over here, it is transporting the solute as well as it can break down ATP to ADP and PI. Got it? Hence it is called as an enzyme. Hence it is called as an ATPase enzyme. Ase means breakdown. Okay. Now this particular transporter protein or enzyme was discovered by Jensko in 1957. You can expect question objectives over here. So remember, this was discovered by Jensko in 1957. Now what this particular transporter does? This transporter is going to couple. Couple means to combine two things. Okay, what two things are combined? First, breakdown of ATP. Okay, पहला क्या काम होने वाला है? First thing is breakdown of ATP. आपका ATP breakdown होगा to form ADP and PI. This was the first thing. And what is the second thing? Second thing is a simultaneous movement of Na plus and K plus against their electrochemical gradient. फर्स्ट क्या करेगा Na plus K plus ATPase, ATP को ब्रेक डाउन करेगा, और दूसरा क्या करता है, वो Na plus और K plus को मूव करता है अगेंस्ट इलेक्ट्रोकेमिकल ग्रेडियंट्स. अगेंस्ट इलेक्ट्रोकेमिकल या फिर अगेंस्ट कॉन्सेंट्रेशन ग्रेडियंट का ये मतलब होता है दैट फ्रॉम लो कॉन्सेंट्रेशन टू हाई कॉन्सेंट्रेशन. इट इज ऑल्सो कॉल्ड एज अपहिल ट्रांसपोर्ट. UPHILL, अपहिल मतलब हिल के ऊपर चढ़ना, नीचे से ऊपर. ओके, इट इज कॉल्ड एज अपहिल ट्रांसपोर्ट बोलते हैं. और जब हम पैसिव ट्रांसपोर्ट और फैसिलिटेटेड का जो एग्जांपल था जिसमें आपका ट्रांसपोर्ट होता है फ्रॉम हाई टू लो, उसको बोलते हैं अलोंग, और अलोंग द कॉन्सेंट्रेशन ग्रेडियंट और टूवर्ड्स द कॉन्सेंट्रेशन ग्रेडियंट, या फिर उसको डाउन हिल बोलते हैं. डाउन हिल मतलब हिल से नीचे उतरना, मतलब हाई से लो जा रहे हैं, इसलिए उसको कहते हैं डाउन हिल. तो ये सारे डिफरेंट टर्म्स है जो आपको डिफरेंट बुक्स में मिलेंगे. फाइन, बट ऑल दे मीन द सेम. ओके, so here since it is active transport, it has to transport against concentration gradient, that is from low to high. Now moving ahead, for each molecule of ATP converted to ADP and PI, मतलब जैसे कि आप वन मॉलिक्यूल का ATP जो है उसको ब्रेक डाउन करेंगे to ADP and PI. So we understood there are two functions. There are two functions or two process is coupled by this particular Na plus K plus ATPase. Na plus K plus ATPase क्या करती है, दो प्रोसेस को कंबाइन करती है. फर्स्ट है ATP का ब्रेकडाउन और सेकेंड है Na plus और K plus का ट्रांसपोर्ट. That we understood, right? Now for each ATP molecule converted to ADP and PI, we have two K plus ions moves inward, while 3 Na plus ions moves outward. जैसे ही आप वन ATP molecule को break down करेंगे to ADP and PI, so as you can see here, we have ATP here. ठीक है, you can see the ATP, and this ATP is getting broken down to ADP and PI. So as one molecule of ATP breaks down to ADP and PI, you have two molecules of K plus. Let us see this here. Two molecules of K plus which is transported inward, means it is transported inside the cell from outside. बाहर से two K plus molecules is transported inside the cell, while 3 Na plus, as you can see here, 3 molecules of Na plus is transported outward, means it is transported outside the cell. Okay, so this is how your Na plus K plus ATPase works. With a breakdown of one molecule of ATP to ADP and PI, we have two molecules of K plus entering the cell and 3 molecules of Na plus leaving the cell. Got it? This is the main important function of your Na plus K plus ATPase. ये जो Na plus K plus ATPase enzyme है, it is an integral protein. We have learned, right? Proteins are of two types, integral and peripheral. So it is an integral protein, means it is embedded in the membrane. आपकी membrane में embedded है. And then we know that these particular Na plus K plus ATPase is having two subunits. It is also a globular protein where it is having two subunits, both of which span the membrane. Span the membrane matlab it pass through the membrane. Okay, it has a outer domain, it has an intracellular domain, it has a transmembrane domain. So we have learnt about all these things previously. Okay, so this particular slide is understood, everybody. Na plus K plus ATPase का main function is clear to all of you.